Nggak terbuka, Bu.
Kak hidup kan negara kak ya lampuan negara ya. Gawin, sudah ibu itu bahas sama rumah. Nggak akap-akap lalu. Baik. Apa ibu ada nyapu itu kotornya ya.
Ya, Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi.
Waalaikumsalam warahmatullahi wabarakatuh.
Selamat pagi juga Ibu.
Salam Aidilfitri untuk semua ya.
Terima kasih Bu.
Okey, alhamdulillah kita dapat bersua lagi pada hari ini. Wassalam.
Masih muda ni.
Masih muda juga. Arah tu yang masih goyang. Mau tumbuh.
Sebentar ya. Boleh nampak skrin saya ya?
Sudah Bu.
Okay, so today we are going to continue. Kita akan sambung lagi perbicaraan kita ataupun perbincangan kita, our discussion about industrial microbiology. So I hope you Assalamualaikum. Mohon maaf ya, ada tiba-tiba internet connection berhenti. Okay. Ya, alhamdulillah ada 17 orang hari ini ya yang akan bersama-sama untuk kita sambung perbincangan kita mengenai industrial microbiology. Cuma saya kurang sihat sikit hari ini, sesama dan batuk. So, kalau kurang jelas suara saya, mohon alert saya ya.
Baik Bu.
Okay, so we are going to continue on our discussion about industrial microbiology. We already discussed about industrial microbiology. I already introduced some microorganisms that involved in industrial in industry, means microorganisms that can be used or as the working organisms to produce products in our industry. Today, we will continue still about microorganisms and industry, but we are going to focus on pharmaceutical products, and also some secondary and primary metabolite, and also how microorganisms involve using DNA recombinant technology in order to produce better products. So basically, okay, we have microorganisms. Okay, normally in their normal day, in their normal role, okay, as biological organisms, they will produce natural products. Okay, so from the natural products, with their potential, we apply them in industrial microbiology. So, as I mentioned earlier or previously, industrial microbiology involves food products, feeds, fine chemicals, recovery of materials, production of biofuels, microbial biomass, single cell protein, metals, biocontrol agents, enzymes, proteins, biopolymers, antimicrobial, cosmetic, and treatment of organic waste, and so on. So microbe can involve in this kind of applications. So we use them, we apply them in order to get better or give benefits to human. So normally microbes produce primary metabolite. Okay, means from their cells they produce this kind of metabolites. Okay, such as vitamins, ethanol, aceton-butanol, or citric acid, acetic acid, and lactic acid. They can produce it, and at the same time, they also can produce secondary metabolite, okay, which also are natural products such as biomaterials, biosurfactants, bioflocculant, and biopolymers, as well as nanomaterials, antimicrobials, symbiotic, and also biofilms. Okay, means they can involve in production of these two kinds of metabolites. So now let's move to the examples of microorganisms, and I will explain. Okay, according to I divide them into discussion on about I divide them into pharmaceutical or related to medicals, bioconversions, and also DNA recombinant. So we typically use microorganisms, okay, in a large scale in order to produce products, okay, and we also use microorganisms to carry out chemical transformations. Certain microorganisms such as fungi or bacteria, we can use them with alcoholic fermentation process, and then later on it can involve process to produce pharmaceutical, food additive, enzymes, and also chemicals. Okay, so basically, if we refer to history, they start they know the potential of microorganisms in alcoholic fermentation process, and with that potential, they explore in order to produce pharmaceutical, food additive, enzymes, and other chemicals. Majority use fungi and streptomyces because these two organisms they have their own varieties, okay, many species, and can produce products. So the process starts with to isolate the microorganisms from environments, in normal environments, and they select for high yielding microbial variants. Means they isolate, they know the potential, okay, they produce in high amount in proper condition, so the microorganisms will produce their products according to our needs. So when we successfully isolate the microorganisms, okay, and we know their potential to produce any kind of products, tidak boleh nampak skrin saya je. Bentar ya. Sekarang bisa nampak skrinnya?
Sudah, Bu.
Okay, let's respond. So just now, I showed these slides. Okay, means the microorganisms involved in industry, as they can produce primary metabolite and secondary metabolites, such as other products from the microorganisms. So I mentioned just now that normally we grow in large scale, and from the history, they start with alcoholic fermentation processes, and later on continue with the productions of other products such as pharmaceutical, food additives, enzymes, and chemicals. So we use majority of fungi and Streptomyces because fungi and Streptomyces are more robust, means they can stand with high temperature, and we can easily manipulate them according to human needs. Okay, so normally we start with isolation of microorganisms from our environment, and then continue with selections of microorganisms that can produce the products. Okay, and we normally choose the high yielding microorganisms. Okay, and when we isolate, either we can identify it first, or we screen their potential first. But beside that, what are the properties that we also screen in order to use microbes in industry? Okay, the first one is it can produce spores or can be easily inoculated. So that's why they use a lot of fungi, because most fungi produce spores, okay, and spores can easily store, okay, to produce as the stock, culture stocks, and use them as working organisms, or bacteria that can be easily inoculated, means with common medium, okay, without using expensive medium, they can grow happily. Okay, normally we will use them because we do not want to use expensive or tedious process in order to get a better product. If we have choices, we will use the organisms that can easily inoculated using low cost. Maybe I can share. There is one group of researcher in UNICEF. They want to grow algae. Okay, so instead of using chemicals or expensive chemicals or ingredients, they try to find natural ingredients from soil, okay, as the medium to be added in their medium to make sure the algae can grow, or the algae can bloom, and from there they will select proper algae to produce other products. Okay, and then second properties is they grow rapidly on a large scale in expensive medium, as I mentioned just now. Means within 24 hours we can get a large amount of cells, okay, instead of using tons of or high volume of medium that can produce low amount of cells. From my experience, my group working on Rhodococcus bacteria, Okay, this kind of bacteria has a lot of potential in industry, but the drawback is this bacteria cannot grow rapidly. Maybe we're still investigating how to grow them in large amount rapidly. Okay, because their character is they only means if within 24 hours you cannot see its bloom in we cannot get large amount of cells. Okay. So normally in industry they will choose the growth rapid bacteria or fungi, and they can grow in large scale in inexpensive medium. And then the third properties is they can produce desired product quickly. Okay, they can produce desired product quickly. We don't want after 2 weeks we will get the the products. If they can grow in 24 hours or 48 hours, it's better. And should not be pathogenic, of course. We do not want to use pathogenic microorganisms in industry because it will affect the worker, and also we don't want it will affect the products, or we don't want to add on other process to get rid the pathogenic effect. And the last one is amenable to genetic manipulations. Okay, we will choose this kind of microorganisms if we know that we can manipulate or we change the character in order to get better products. So this is among the microbial products of industrial interest. The microbial cells itself, okay, the enzymes, okay, antibiotic, steroid and alkaloid, food additive, and commodity chemicals. Because using microorganisms, it can produce inexpensive chemicals produced in bulk, which include ethanol, citric acid, and and others. Okay, now we move to now you already know the character of microorganisms that normally used in industry. Now let's discuss on the production and scale. Okay, we divide into two types. Okay, first one is primary metabolite and also secondary metabolite. For primary metabolites means it produce first, okay, and normally they produce this kind of products or metabolites during exponential growth. Whereas for secondary metabolites, they produce in stationary phase. Before I go further, have you ever heard these phases? Exponential, stationary, the lag phase, log phase. Pernah dengar dalam proses pertumbuhan microorganisma? Ibu bapa pernah dengar sebelum ini? Rasanya pernah dengar ya dalam kuliah pertumbuhan microorganisms. Saya percaya sudah dengar.
Ya, Bu.
Okay. Jadi, so the primary metabolite normally produced during exponential growth, means during their active phase, and secondary metabolite during the stationary phase. And normally, what happened for primary metabolite? Okay, they produce and certain products, they use it in their growth. Whereas for secondary metabolite, okay, it's not essential for their growth. Means it's a side. Okay, products. Actually, it's not important for them, but they can produce them. And normally they produce or it form the products depending on the growth conditions. Okay. And they produce as a group of related compounds. Okay, it's not one, but they have related compound, and it often significantly overproduce. Okay, they can overproduce because it's not important, but when they're happy to produce, they can produce. And it's widely produced by spore forming microbes during sporulations. Because microbes, when they grow, okay, when they become matured, in order to survive or in a harsh condition, microbes can produce spore. So secondary metabolites produced during sporulation. Okay, this figure shows you products, the productions of microbial products versus time. Okay, for A is for alcohol, sugar, or cell number. You can see as when they grow, they have the primary metabolite. Okay, so it's important for their growth. So the cells, it can produce more cells, it will produce more alcohol. Okay, but the sugar in when they will use sugar during their growth. For penicillin, sugar or cell number, okay, you can see here the amount the penicillin produced as secondary metabolite. Okay, when the you can see when the cells at stationary phase. All right. Compared to primary, you can see it produced during exponential phase. Right. Normally, for secondary metabolite, okay, they produce large amount of organic molecules, and we cannot get it straight away, but it involves many steps and use specific enzymes. Okay, for example, synthesis of tetracycline, okay, this kind of antibiotic, it requires at least 72 separate enzymatic steps. Okay,
This is yours.
can you imagine a lot of enzymatic steps? And the starting materials arise from major biosynthetic pathway. Okay, so it start with a common materials or common biosynthetic pathway, and it will continue later on. So for production, it use in industry use fermenter, okay, where all the microbiology process take place, and most are using aerobic process. Fermenter can vary in size, okay, starting from 5 liter, this is a minimum fermenter, up to 500 liters. When they build the fermenter, okay, they can set either the process will be used or will be going on aerobic or anaerobic conditions. And if you have chances to go to industry, or you can find any resources, okay, you can see that large scale fermenter are almost or always made up by stainless steel. Okay, and they have impellers and sparges to supply oxygen. Okay, this is a small fermenter. Okay, about 5 liter, and normally they will attach to a computer because we want to monitor the conditions. We want to know the oxygen, the carbon dioxide, and everything. So it depends, the parameter depends on the type of microorganisms and also the types of products. This is the crosscut of fermenter. Okay, so normally there is one inlet that supplies sterile air, okay, the oxygen, and then it has sparger, okay, to make sure the air can be blown in the entire fermenter. So we will add the culture growth. Okay, you also can see the impeller here to make sure it will mix the culture growth throughout the process. Okay, and normally it has cooling jacket, okay, to avoid, of course, when there is mixing, all right, and the bacteria is growing, it will produce high temp, not high, but it become high temperature. So that's why they need cooling jacket, and so they have also external cooling system. All right, and then normally they also have viewing pod to observe. All right. And it has pH meter to know the pH, to monitor the pH, right? And also they have few pumps, okay, according to the needs or the productions, right? So from time to time, normally they will get samples to monitor the products, okay, and also the conditions, the culture condition. When the process is complete, they will harvest the products. So this is a large fermenter where you can see here. It's a large fermenter. This one used by an enzymes company, okay, Novo Norzymes or Novo Nordics. Okay, it's a large fermenter up to 500 liters. Right, so they use normally in industry not only one fermenter but large amount of fermenters. So it depends on the industry. Okay. Any questions until or up to this point?
Iya Bu, izin bertanya Bu.
Ya.
Baik, tadi terkait dengan slide mengenai properties of a usable industrial microba includes, di situ tadi dikatakan bahwa untuk pertumbuhan skala besar bakteri itu kita harus menggunakan salah satunya adalah medium yang inexpensive medium. Oke. Pertanyaannya, please provide an example of medium.
Oke.
Bisa ya untuk medium itu sendiri Bu. Kemudian di sini berarti dikatakan bahwa ini merupakan karakteristik mikroba yang berpotensi sangat baik Bu ya untuk produk bioteknologi. Apakah seperti itu Bu?
Ya betul.
Oke.
So maksudnya di sini of course kalau kita boleh dapat produk yang sama tapi menggunakan medium yang lebih murah berbanding kalau kita dapat amount yang sama tapi ataupun amount yang lebih besar tapi menggunakan medium yang lebih mahal. Contoh yang murah dan yang mahal ini, yang murah ni biasanya kita menggunakan yang semula jadi, yang natural. Okay? Berbanding yang kita perlu tambah vitamin, kita perlu tambah trace elements. Biasanya trace element ni kita guna dalam jumlah yang sedikit tetapi dia banyak jenis. Banyak jenis dan pastinya dia akan memberi ataupun meningkatkan kos yang lebih tinggi. Sebab itu untuk proses awal, untuk mengetahui potensi, yang normally kita akan tahu potensi microorganisms itu, maybe kita bakteria ini dia boleh menghasilkan tetracycline contohnya, antibiotik. Tetapi dan kita akan cuba menggunakan medium-medium yang biasa. Contoh kalau dalam screening proses kita akan menggunakan nutrient agar, di mana dalam nutrient agar tu hanya ada gula dan juga starch. Contoh Okay, dan yeast extract for example. So dia tidak terlalu mahal berbanding dan kita akan cuba hidupkan dia jika dalam medium yang sebegitu, yang murah itu dia boleh menghasilkan produksi yang bagus, kita pilih yang itu. Berbanding kalau kita letak vitamin, trace element dan sebagainya, tetapi dia juga boleh menghasilkan produk yang lebih kurang sama. Okay, jadi better kita guna yang lebih murah berbanding menggunakan yang lebih mahal.
Berarti lebih banyak yang medium agar yang seperti biasa kita lakukan, Bu ya?
Iya,
pada umumnya.
Ya, biasanya kita akan mula kerja kita dengan yang itu. Okey, tapi kalau dia tak ada potensi kan, kita boleh tukar, tetapi at the end kita akan tengok jugalah sama ada dia dia menggunakan terlalu mahal dan kita tak akan pilih lah yang tu. Seperti yang saya sebut tadi, ada satu kumpulan penyelidik di Universiti Selangor yang cuba menggunakan tanah yang dimasukkan dalam medium dengan harapan tanah itu akan memberi properties yang baik, yang boleh menggalakkan to increase the amount of cells for the algae. Mereka cuba untuk hidupkan algae, jadi mereka ambil tanah masuk dalam medium, dan tengok mereka pergi cari pelbagai jenis tanah untuk tengok mungkin ada tanah yang boleh memberi properties yang baik dan induce the growth of algae itu sendiri. Adakah saya menjawab?
Oke, terima kasih Bu. Satu lagi boleh Bu?
Ya.
Oke, terkait dengan amenable to genetic manipulation, di situ grup what group of bacteria are amenable, Bu?
Maksudnya dia boleh kita amend dia. Maksudnya kalau kita tahu dia ada potensi, katakanlah dia ni boleh menghasilkan enzim amylase contohnya, tetapi untuk meningkatkan lagi potensi dia, kita cuba ambil gene daripada ataupun DNA daripada bakteria ini, ataupun kita mixkan dengan gene yang lain, contohnya untuk menjadi dia lebih baik menghasilkan amylase tadi.
Jadi biasanya kelompok bakteri apa, Bu?
Contoh
yang lebih amenable itu dari sebanyak mikroorganisme yang digunakan biasanya.
Kalau kita tengok sekarang banyak yang digunakan E. coli, itu antara yang banyak lah, sebab E. coli ni dia sangat mudah untuk digunakan dalam proses genetic manipulation.
Oke. Ya, terima kasih Bu.
Sama.
Iya, semoga Ibu cepat sehat. Kayaknya flu berat Bu ya.
Itulah, sorry kalau suaranya tidak terlalu jelas.
Iya, nggak apa-apa Bu.
Okey. Jika tidak ada soalan lain, kita teruskan. Okay, we continue about antibiotics. How we want to isolate the isolation, yield, and purification. So what is the meaning of antibiotics? Basically, it is secondary metabolite. Okay, it's a product from microorganisms, but we use it to kill or inhibit the growth of other microbes. And currently, most antibiotics in clinical use are produced by filamentous fungi or actinomyces. It starts with, for the history, it starts with the works of, do you know who is the father of antibiotics? Siapa tahu? Siapakah pemula untuk ataupun berjumpa dengan antibiotics?
Alexander Fleming, Bu.
Okey, dia menghasilkan apa?
Penicillin, Bu.
Ya, penicillin. So dia secara tidak sengaja, ya kan, dia nampak bila ada satu bakteria, satu fungi yang hidup dan bersama-sama dalam plates, dan ada kawasan yang telah menyebabkan bakteria itu tidak tumbuh. Alright. So so it start so it need a lab screening process, okay, and normally we obtain from nature, okay, to get the pleo culture, and we assay, okay, we do analysis to see the products or how or what kind of antibiotics that inhibit the growth of test bacteria. Okay, I just want to share one video related how they it's not working just for a while. Bentar ya. Hang pula. Saya cuba untuk share skrin saya semula. Tak mengapa, tapi saya nak cuba, saya mau share slide saya semula tapi masih hang di sini. Saya mahu continue discussion on antibiotics. I want to show my slide actually, but it's not, still not, I think still not appear in front of you ya. Belum ya.
Belum.
Saya mohon maaf kenapa tidak boleh ni. Basically, I want to continue to discuss about antibiotics. So among, that's while waiting, maybe I just continue to discuss to mention about what kind of antibiotics. As you mentioned just now, penicillin is the common antibiotics. It is natural and it can produce natural and also biosynthetic penicillin. It also have semi-synthetic penicillin because it has broad spectrum of activity, and it also secondary metabolites. It's a kind of secondary metabolite, and it requires low amount of glucose, because if high level of glucose in the medium, it will repress the productions of penicillin. For your information, in the biosynthesis of tetracycline is more than 72 intermediates involved, and there are more than 300 genes involved, okay, in order to produce a large number of tetracycline. Okay, besides antibiotics, in industry, they also produce vitamins and amino acid from the microorganisms. And so antibiotics, we can say it is a pharmaceutical product, pharmaceutical products produced by microorganisms. Beside antibiotics, vitamin is the second one, okay, such as vitamin B12. It exclusively produced by microorganisms. And then riboflavin also produced by microorganisms. Besides vitamin, amino acid also as the secondary metabolite, where it used as feed additive in food industry, and also produced in high amount as the nutritional supplement in nutraceutical industry. Okay. And normally amino acid used as the starting materials in chemical industry, okay, such as glutamic acid or MSG, aspartic acid or phenylalanine, or we commonly known as heard about aspartame or the NutraSweet. Okay, means it's not it's synthetic, or lysine as the food additive. Okay, beside antibiotic and amino acid in pharmaceutical industry, we also get enzymes as the industrial products from microorganisms. Okay, if it is exoenzymes, means the microorganisms produce it, excrete it from the cell. Okay, it's produced extracellular, or it also can use intracellular, but we commonly choose extracellular because we want to avoid the process to break the cells. Okay, so enzymes are useful in industrial or as the industrial catalyst. Okay, to speed up the process in industry. Can you see my slide now? So enzymes produce and also used a lot in industry. Okay, next is alcoholic beverages and biofuels such as wine, brewing and distilling, and also biofuels. Okay, as we know, most wine is made from grapes, and they produce in 200 to 200,000 liters fermenter. Okay, so there are various type of wine, white, red. Okay, and normally it depends on the type of grapes and type of microorganisms, as well as the durations of the process. Okay, this is the fermenter for the Christian Brothers Winery. Okay, it's a products that a company that produce wines. Okay, this is a quite traditional. Okay, so in the process, so you can see here are different. Okay, the duration are different, and also depends on the type of grapes. In brewing and distilling, actually this is a terms that used to describe the manufacture of alcoholic beverages from malted grains. Okay, and yeast is widely used to produce the beer, and there are two types of brewing yeast strain. So you can see here, this is kind of a fermenter to produce beer. And normally it involves temperature and use large fermenter for industrial purposes. And from this, they also use as an industrial solvent and also gasoline supplement. Okay, they use the ethanol from the industry to use as a solvent and gasoline. Okay, this is different kind of beer. Okay, beside that, beside wine, ethanol, and biofuels, beside wine and beer, they also produce biofuels, okay, such as ethanol biofuels and petroleum biofuels. Okay, so ethanol produced using fermentation of feedstock, and then petroleum biofuels, they synthesize from green algae. This is a technology or potential of algae that can produce as petroleum. Okay, this is an example of Chris Stendal and DOE and Enreal company. Okay, where they use to produce biofuels. Okay, next, part is about products from genetically engineered microorganisms. They can produce as the expressing mammalian genes in bacteria, or productions of genetically engineered somatotropin or other mammalian protein and products, genetically engineered vaccine, mining genomes, and also engineering metabolic pathway. If you still remember during COVID-19, right? There is a lot of effort they tried to engineer the process of for production of vaccine, right? So that kind of products from genetically engineered microorganisms. So in biotechnology, they use living organisms for industry. For GMO, because normally we want to use the natural organisms, but if we know the potential, we try to alter the genome. All right. So in order to express more, or they allow the expression of eukaryotic genes in prokaryotes. For example, in production of insulins, okay, they get the genes and produce in bacteria. So they will minimize the cost because bacteria can be grown in large scale. So how they get it? They clone. They clone the gene via mRNA, for example, or they find the gene via the protein. Means we reverse. We know the protein, we know the products, then we find the genes and isolate the genes and also clone the genes to alter the organisms to get a better products. This is just a brief about how they get gene from mRNA. This is the common process. I think you will learn it later on at the end of this course under genetic for microorganisms. So as the principal step of a bioengineered methodology process, okay, normally we have upstream process where we prepare the microorganisms and the raw materials, okay, and then followed by fermentation and biotransformation. After that is the downstream, or we want to purify the desired compound or the desired products, okay, from the cell. Okay, either we have to break the cells or we just collect from the medium. So related to genetic engineer, normally we try to create variants, okay, to produce better products, okay, and we improve the strain, okay, to get the products, or we manipulate the genes. Okay, we get the genes and clone it in another other organisms. I give you example just now. For insulin, we get the genes and then we produce in bacteria. So of course, we will get insulin in large amount. Instead, if we kill more cows to get the insulin. So this among the scientists that involved in recombinant DNA technology. Okay. I just want to show okay, so this is the about insulin. It's the first commercial product from DNA technology. Okay, because previously they start with human insulin and then get insulin from to produce in cows. Okay, dalam lembu. Okay, but nowadays the DNA sequence that encodes for the human insulin, okay, then they clone it, transfer or transplant into plasmid, and then this plasmid will transfer to host. The common bacterium is E. coli, so the host will act as factory, alright, and then they produce the human insulin, right? So after that, they are combined and purified and used to treat diabetics. Okay, because previously they get insulin from cows and also majority from pig or porcine, which is quite sensitive for us as a Muslim, right? And also we can avoid to kill porcine in order to get insulin to treat diabetic. So we can get recombinant insulin. We can produce in large amount. It's safe and is halal to be used for the Muslim. So if you can see from this diagram, right? So molecular biotechnology that involve microorganisms, okay, can be used for the benefit of crops, drugs, production of vaccine, genetics, diagnostic process, and for livestock. So this part, I think you will learn later on when you discuss about genetics for microbia by other lecturers. Okay, so as the conclusion, we can see now that microorganisms not only pathogen to human, but it also can be manipulated, or we can use them to the benefit of human, or to be used as the working organism in industry, or we can get products from microorganisms and produce in large scale in industry. So we can see that microorganisms can give us benefit, okay, for a better life, and also to create economy for human to by manipulating the microorganisms and also their products. Okay, so I think that should be all that I want to discuss today. Is there any questions? I open for discussion.
Maaf Bu Azah,
Ya,
nanti boleh PPT-nya dibagi Bu?
Boleh.
Ya Bu, terima kasih.
Saya akan beri nanti. Yang lalu sudah dapat?
Belum
Yang
juga
lalu?
Bu.
Belum diterima ya?
Iya, sekalian aja Bu ya.
Nanti saya
Terima kasih Bu.
Saya mohon maaf dengan technical tadi ya. Saya mungkin the hanging tadi. Tapi saya akan masukkan, dan ibu bapa semua boleh lihat sendiri link-link yang saya beri, dan saya juga sertakan link berkaitan dengan industrial microbiology daripada journals dan bahan-bahan bacaan yang lain yang boleh dijadikan rujukan dan memberi maklumat lanjut lah berkaitan apa yang kita bincangkan hari ini. Ada soalan lagi? ke belakang apa ya, Ibu Siti Nurhayati? Okay, sebelum tu, I just want to share a few pictures related to products from microorganisms. Okay, so in different countries, we have different products that use microorganisms, okay, in the productions of foods, especially. Okay, this is ester that use immobilized lipase from yeast, Candida cylindrica. Okay, they produce esters. This is diacetyl, the conversion of citrate by Streptococcus diacetyl-lactase, and also Leuconostoc citrovorum. Okay, they induce citric acid in permease system. This is pyrazines. They culture Bacillus subtilis, okay, to produce tetramethyl pyrazine. This is lactone. And then that normally used for baking, right? They involve Candida and Saccharomyces, Penicillium notatum, Cladosporum, okay, and Sarcina, and also Cladosporum suaveolens. Okay, they use it, and then terpenes. Okay, you can see they use a different kind of microorganisms here. Okay, in production of flavor, okay, MSG, glutamic acid. This is 5' nucleotides. They use fermentation of Bacillus subtilis, Brevibacterium, and Actinomyces. Beside that, production of color, okay, that use from fungi or mold. This is color from yeast, okay, to produce astaxanthin, dia macam warna kemerahan. This is from algae, okay, phycocyanin and phycoesterine is a part of chlorophyll A, is green in color. And also Cyanophyta and Chlorophyta algae that produce red and blue pigments that known as phycobilipotins. Okay, so you can see this is a natural color. So it will be safe for us compared to synthetic color. Okay, apa tadi soalannya? Tadi di sini juga saya sertakan link ini ya, boleh baca. Ibu Siti Nurhayati, ingin saya tunjuk yang mana?
Tidak terlihat.
Apa dia?
Tidak terlihat.
Berkaitan dengan?
Tidak ada yang terlihat besar.
Okey, ada soalan ke? Jika tiada apa soalan, mungkin kita boleh berhenti di sini.
Baik Bu, cukup Bu.
Cukup ya.
Ya,
Mungkin ini hari Sabtu. Di sana masih berhari raya ke?
masih Bu. Masih ada keluarga.
So hari ini mungkin ada yang mau keluar untuk berhari raya kan. So saya pun sebentar lagi ada tetamu ingin datang beraya.
Ya Bu, cepat
Okey.
sehat Bu.
InsyaAllah. So terima kasih kepada semua. Mohon maaf andai ada salah dan silap, especially ada technical issue. Dan jika ada apa-apa soalan ataupun maklumat yang ingin disampaikan kepada saya, boleh disampaikan kepada pihak penyelaras, koordinator, dan nanti insyaAllah saya akan kongsikan slide ini melalui mereka ya.
Baik Bu.
Okey. Terima kasih semua.
Sama-sama Bu.
Okey, Assalamualaikum warahmatullahi wabarakatuh. InsyaAllah kita jumpa lagi bila-bila.
Amin.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Sama-sama.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Gimana kabarnya? Semua sehat?
Alhamdulillah.
Alhamdulillah sehat Bu. Ibu
Nah.
Yeti gimana Bu?
Alhamdulillah sehat. Ada pendatang baru ya?
Ya Bu, siap Bu, ada Bu.
Ibu Kun ya?
Ya Bu.
Yati. Oke, selamat datang, selamat bergabung Ibu Kun.
Ya siap Bu.
Ini apa? Kamera saya kayaknya ada masalah ya. Jadi agak hijau-hijau terlihat enggak?
Iya Bu.
Iya, jadi kayak nggak ngerti saya settingnya apa memang ininya bermasalah. Ya udah mudah-mudahan tidak mengganggu ya. Hanya kayaknya pucat ya bibir saya itu lucu jadi kelihatannya. Benar enggak? Saya nggak tahu nih settingnya gimana. Sebentar. Saya cek dulu. Tapi nggak ada filter background ini.